Assalamualaikum. We are from Group 1. We are going to present you about course AQU 239 Larva Food Production. First of all, let me introduce our group members. We have Nurul Izzati, Rafiq Kuhairi, Akmal Akamaluddin, and the last one is me, Nur Humaira. This is our table of content, and our title is about the effect of different media in the growth of algae. Our objective here is to make sure microalgae started for the next culture in the next stage. Next, to prepare fish meal to be tested as the media, to gain initial number of microalgae cells, to compare performance of NPK fertilizer and fish meal in alga growth.
First of all, I'm going to introduce you what is microalgae. Microalgae are vital in aquaculture as a primary food source for larvae fish, shellfish, and crustaceans, providing essential nutrients like proteins, lipids, and omega-3 fatty acids. They enhance water quality by recycling nutrients and stabilizing the ecosystem in aquaculture. Additionally, their use improves the growth, health, and survival rates of cultured species, making them indispensable in sustainable aquaculture practices. Below, I have provided a picture of algae culture and a short video of the process in algae culture.
Introduction of NPK fertilizer. NPK fertilizer is commonly used in algae culture as a source of essential nutrient, nitrogen, phosphorus, and potassium to promote algae growth. Nitrogen will support protein synthesis. Phosphorus is crucial for energy transfer and cell division, and potassium enhance overall metabolic process. Proper dosing is important to optimize growth while avoiding a nutrient imbalance or water quality issue. Introduction of fish meal. Fish meal is used in algae primarily become as nutrient source, provide an essential protein, amino acid, and mineral that will support the growth of the microalgae. It enhances the culture medium by supplying the organic nitrogen, which is promote a high-density algae growth. However, it must be careful in manage it to prevent from contamination and maintain a good water quality.
Before doing an algae culture process, we must know how to prepare the algae starter. I'm going to explain to you the preparation of algal starter. Firstly, preparation samples of water to be examined under microscope. This is to make sure the presence of algae. Next, examine the water samples that have been taken under the microscope to ensure the presence of algae itself. The water samples that have been examined and have been confirmed that the water samples consist of algae. This is the short video of our group identifying an algae. Next, we must prepare the materials for algae starter process, which is we need 4 water bottles, a pipette, and a water samples of algae. Next, we're going to fill in the half of the water bottle with distilled water, and then place a couple drops of nutrient-rich solution into the water bottle that have been filled with water. Place the water samples that are examined with the presence of algae into the water bottle that have already been prepared. Gently shake the bottle for 1 minute so that it is well combined and no spillage occur. Figure 9 showing the results after the preparation of algae starter.
Preparation of fish meal. First, thaw the fish first and make sure the fish not frozen. Second, ensuring the fish is in a clean condition. After cleaning, arrange the fish on the steel tray. Place the fish in the oven. Set the oven to 45 Celsius with 100% fan speed. Process of drying fish for 1 week. Turn the fish upside down during the drying process to prevent moisture in fish still exists. After that, after 1 week of fish drying, set the fish aside. Then grind using electric blender. The result of fish meal after blended. Next, put it back in the oven to ensure the fish meal is completely dry. Lastly, keep the fish meal in sealed plastic with proper label.
When culturing algae, counting algal cells are very critical aspects in order to make sure the production of algae are increasing or deteriorating. I'm going to explain to you how to count algal cells. First of all, clean the hemocytometer and the cover slip with alcohol to ensure a clear view. Place the cover slip on the hemocytometer, ensuring it sits flat and aligned with the groove. Next, mix the algal culture thoroughly to ensure an even distribution of cells. Use a pipette to draw a small amount of algae sample. Gently load the sample into one of the counting chambers by placing the pipette tip at the edge of the chamber. Position the hemocytometer on the microscope stage and focus on the counting grid using the low-power objective lens. Adjust to higher magnification if necessary. Next, we have to identify the grid on the hemocytometer. Next, select a counting area, 4 corner squares, and the center square of the grids, and the count of the algal cells within the squares. Count cells in a multiple grids or repeat the process in the second chamber of the hemocytometer for more accurate result.
For the next method, putting media into treatment 2 and treatment 3. First, take a fish meal that has been packed. Second, measure the NPK fertilizer and fish meal to put into the tank treatment 2 and treatment 3. Next, NPK fertilizer which has been measured and placed into tank treatment 2. And lastly, fish meal which has been measured and placed into tank treatment 3. Result on initial algae cell. Initial algae cell on treatment 2, which were using NPK fertilizer, shows the best result compared with the treatment 1 and treatment 3. However, treatment 1 hold the algae counting lowest data between all of the treatment.
Result of algae cell in week 1. On week 1 of counting algae cell, it is shown that treatment 2 while using NPK fertilizer is the most effective treatment. However, algae cell treatment 1 which not using anything are increasing rapidly more than treatment 3 fish meal. Result of algae cell in week 2. On week 2 of algae cell counting, it is shown that treatment 1 has significantly increased and is the most effective treatment for week 2, past more than treatment 2 and 3. Meanwhile, treatment 3 is algae cell are increasing more than treatment 2 in week 2. Result of algae cell in week 3. In week 3, we can see that treatment 3, which using fish meal, increased rapidly and abrupt the treatment 1 and treatment 2. Meanwhile, treatment 1 are significantly decreasing until it's become the most ineffective treatment on week 3 and hold the algae count the lowest between all of the treatment.
As for the final result, we can observe that T3 has the highest data collection between T1 and T2. Fish meal causes more algae growth than NPK fertilizer because of how it releases nutrients. As it breaks down in water, it adds nitrogen and phosphorus which algae readily use. Fish meal releases nutrients slowly over time, keeping algae fat for a longer time. Since it is organic, it also helps in decomposition, making more food for algae. NPK fertilizer release nutrients quickly but don't support algae growth in the same way. Based on the result, we can know the treatment 3, which is fish meal, are the best for growth performance to the algae.
Challenges that we face is uneven light distribution. As we know, microalgae require optimal lighting minimum 18 hours to carry out photosynthesis and produce energy. Uneven light distribution can reduce amount of light that microalgae receive. It is better to use light spectrum like blue and red light. Blue light can bring chlorophyll formation while red light produces the extra energy needed for photosynthesis. Next, the problem of contaminated water with insects with big impact on microalgae. Insects disrupt the microalgae ecosystem because it may bring bacteria or pathogens, and insects change the pH and temperature that damage growth of microalgae. So it is take time and energy to clean the insects that are present. And the last one is aeration must be maintained all the times to prevent the sedimentation of algae, ensuring algae do not crash and avoid thermal stratification.
Next is future direction of the study. First of all, we can create alternative ingredients for feeds, such as insect-based feed, investigating the potential for insects, specifically black soldier fly larvae, as an alternative source of protein and lipids in larval diets. Other than that, it is plant-based solutions. Investigate plant-based solutions like soy, algae, or other new protein sources and their effects on the health and growth of larvae. Next, we can also learning about nutritional science, which is profiling micronutrients deficiency in typical larvae diets and their impact on growth and survival. In addition, interaction of feed with the gut microbiome in larvae associated with health and resistant to disease, exploiting the potential of addition of probiotic or prebiotics to larval diets through improvement of intestinal health and immunity. Next slide, I'm going to introduce you the implication of the study. From what our class have learned, algae is one of the most challenging species of microorganisms to culture because of so many risks that can absolutely occur when there is a slight mistake and error, which then can lead to algae bloom, such as pollution, improper handling, and also incorrect temperature. However, by this knowledge, we can use them to be wise aquaculturists that emphasize preventing overtreating in order to gain more profitability in this particular study. From algae culture project that we have conducted, our group definitely gained both theoretical and practical expertise in cultivating, harvesting, and counting algae effectively in aquaculture. As an aquaculture student, this knowledge is very critical for supporting the larval stages of aquaculture species and also enhancing sustainability and creating efficient production system.
The summary is choice of media important in determining how well algae grow, and by adjusting factors like nutrient composition and others in this slide can optimize the growth of algae.
So this is our group references, and that's all from our group. Thank you.